Hello, and welcome to the Physics World Weekly Podcast. This episode features the Nobel laureate Ferenc Krausz in conversation with Physics World's Tami Freeman. Krausz explains how ultra-short-pulsed laser technology could provide a diagnostic tool for early disease detection. They chat about the Science for People initiative, a charity that Krausz and colleagues founded to provide education for children who have been displaced by the war in Ukraine. And Krausz also talks about his collaboration with Hungary's Semmelweis University to establish the John von Neumann Institute for Data Science.
In 2023, the Nobel Prize in Physics was awarded to Ferenc Krausz, Pierre Agostini and Anne Rouillier for their research on attosecond pulses of light. Ferenc Krausz is director of the Max Planck Institute of Quantum Optics and a professor at LMU Munich in Germany. He's also CEO and scientific director at the Centre for Molecular Fingerprinting in Budapest, Hungary, where he is using ultra-short-pulsed laser technology to develop a diagnostic tool for early disease detection. And earlier this year, Ferenc received the Semmelweis Budapest Award from Semmelweis University in Hungary for his groundbreaking contributions to science. I'm very pleased to say that Ferenc has joined me today to discuss how attosecond physics is poised to revolutionize healthcare. Welcome to the podcast, Ferenc.
Hello, thank you for having me.
So, your research team is developing a diagnostic technique called infrared electric field molecular fingerprinting. Can you explain how this works?
Sure. I can try to do this in a few sentences. Basically, the basic idea is to excite molecules with a very brief infrared laser pulse. This brings the molecules into vibration, and the vibrating molecules emit infrared light that we can capture with the help of ultra-short laser pulses. These signals depend on the molecular composition of the sample, which is supposed to change whenever the physiological state of the organism is changing. Whenever a disease is about to develop, usually the molecular composition of blood is changing, and this very signal that we have dubbed the infrared molecular fingerprint of the sample is changing, and it is this change that we are after.
Okay, so you and your colleagues were the first to create attosecond light pulses. So how does this use of this short pulse laser technology enable the new diagnostic tool?
Well, it's not directly the attosecond pulses that we actually use here. So that's I think an interesting story, which also shows how purely curiosity-driven research eventually can spawn some application that's maybe useful even basically for everyone in basically in real world. Actually, the attosecond technology that we have developed eventually enabled us to not just capture the motion of electrons, which was the original purpose, which was the original motivation for generating and measuring attosecond pulses, but they also allowed us to actually capture the oscillation of the electric field of visible light. And it is this very capability that led us to the idea, what if we could use this technique for capturing the light that these molecules, these excited molecules send out once they are excited by very short infrared laser pulse. So it is basically the ability to capture, to sample the electric field oscillations of light that opened the door this new application in medical diagnostics.
Okay, and could you perhaps give an example of how this molecular fingerprinting could be used to detect early signs of cancer, for example?
Well, this is exactly what we started to do originally about 10 years ago. We teamed up with clinicians who collected blood samples for us, blood samples from cancer patients on one hand, and blood samples also from control individuals. To compare the samples from the cancer patients with. And we transilluminated all these samples with our infrared light and measured these infrared fingerprints and figured out that indeed the infrared fingerprints from the blood samples of cancer patients looked significantly different from those of the control individuals. And these differences could be used and can be used to actually train an algorithm to recognize these differences by the use of artificial intelligence. And the approach is that we take basically a certain fraction of the collected samples, both from the cases and controls to train the algorithm, and use the remaining part of the samples, of the collected samples, to test the algorithm. And it was this procedure which allowed us to actually figure out that indeed the infrared fingerprinting is able to detect with significant or with very promising efficiency differences in the infrared fingerprint between lung cancer patients, for instance, as compared to either healthy individuals or individuals suffering from another disease of the same organ, in this case lung.
So you're measuring differences in this infrared fingerprint. So is that just the spectra from different molecules in the blood sample? So is it the composition of the sample is different, for example, in a cancer patient than in a normal blood sample?
Exactly. The composition, the molecular composition of the sample happens to be significantly different, and this manifests itself in differences in the infrared signals that the excited molecules of the sample send out, which we can capture.
So you've got some ongoing trials running, looking at blood samples. How are these progressing?
We started actually on lung cancer, and this is also the type of cancer that we have investigated most extensively so far. We have collected in cooperation with clinicians from the clinics of the University of Munich, thousands of samples from lung cancer patients and from control individuals. And it was just recently that we published these results showing that we can detect this disease at a stage when the cancer is still localized, not metastasized, which is very important because this allows a much better prognosis with quite a reasonable, promising efficiency, which motivates us to move on and work on advancement of this technique. This also motivated us actually to extend the study, and meanwhile, we have looked also at several other cancer entities. Altogether at 8 different types of cancer entities so far, and in all cases we were able to detect significant differences in the infrared signals from cancer patients with respect to control individuals. This actually encouraged us to go even beyond and look at other common conditions like prediabetes, diabetes, COPD, and also cardiovascular conditions. And in all these cases, we have found in preliminary studies very encouraging signs of patterns that we can actually, we means in this case again artificial intelligence, can recognize and distinguish from the signals coming from the samples of the control individuals. So this actually motivated us to set ourselves a bigger goal than just focusing on individual diseases. And we asked ourselves whether this very method could potentially be suitable for doing a comprehensive health monitoring. Basically, we asked ourselves whether this very method could potentially serve as a cornerstone for a comprehensive health screening program where individuals come once a year, donate blood. Their blood is analyzed with this technique, possibly also with some other techniques like proteomics or metabolomics. And the molecular information that we pull out from these blood samples could result in a reliable assessment of the health state of the individual. So this was the question that we posed several years ago, which led us to initiate a large-scale clinical study in Hungary, which we dubbed the Health for Hungary, Hungary for Health study, in the framework of which we enrolled initially some 10,000 individuals, and now we are about to enrolling another 5,000, so that altogether 15,000 individuals are participating in this study, who are going to be followed up over a period of 10 years with frequent blood sampling. In the first year, they are recalled several times so that within a short period of time, we can establish their individualized baseline. Basically, the individualized baseline fingerprint. This is different for each and every person, just as our true fingerprints are different. And therefore, it needs to be established for each and every individual. This happens in the initial period by this repeated blood sampling. And once this is established, from then on, we just follow them up year by year and compare the newly measured fingerprint with this baseline. And if the newly measured fingerprint is kind of within the uncertainty of this baseline, which we can assess from the initial repeated samplings, then we say, okay, nothing has changed. And obviously, based on this fingerprint, there is no severe conditions about to emerge. And if we see that there is some change, then we actually try to correlate this with the conditions that are eventually being diagnosed by standard of care. So basically, in this study, we wish to test and validate this method as a potential method for comprehensive health screening for the most common chronic conditions like cancer, cardiovascular diseases, respiratory and metabolic disorders.
I mean, that sounds really fascinating. So someone could just go and give a sample of blood and it could be tested for all these different potential diseases. But you mentioned that it's your technique looks for changes in these fingerprints. So if someone were going for health screening, would they need to give this baseline sample at some point when they're sort of relatively healthy, and then they'd just be followed up throughout the years? Is that how you could see it would work?
This would be the ideal situation, exactly. That we start this at a stage when people are still healthy, and then establish their healthy baseline, and we then later on measure and look for possible deviations that would come up with respect to their healthy baseline if some new onset disease is about to come into play.
Okay, and you mentioned that you're using artificial intelligence to analyze, I guess, the massive amount of data that you're recording. Has the use of AI, has that really helped you enable you develop this technique?
I would say it's not just helped us, but it's a kind of enabling technology here. It is as important as the physical methodology itself, because the differences in these infrared signal patterns between the diseased and healthy individuals are so subtle that it would be very, very difficult and challenging for our human intelligence to actually reliably assess and evaluate these differences. So here, artificial intelligence is really a true game changer, actually an enabling technology.
Okay. Now, at the end of last year, the Centre for Molecular Fingerprinting and Semmelweis University set up a joint research institute called the John von Neumann Institute for Data Science. Um, can you just tell me a little bit about this and what are the aims of this new institute?
Yeah, first of all, maybe a couple of words about the Center for Molecular Fingerprinting. This is actually the institute that we initiated about 5 years ago. Actually, meanwhile, almost 6 years ago, which rolled out this clinical study that I have been talking about, this Hungary for Health study, and um, as I just mentioned, a very important component of the research at this institute is the analysis of the measured fingerprints and possibly other molecular data that we extract from the collected blood samples via artificial intelligence. And therefore, data science is an absolutely cornerstone and key component in this research endeavor. And we thought that joining forces here with one of the most internationally known medical universities in the Central European region, with Semmelweis University, will be mutually beneficial to advance this technology, eventually to the benefit of the people, of course. And that's why we decided to actually establish this joint institute. And under the umbrella of this institute, we wish to pull together researchers from CMF, from Center for Molecular Fingerprinting on one hand, and from Semmelweis University on the other hand, who share a common interest in utilizing data science and utilizing artificial intelligence for tackling the medical challenges.
Excellent. So the new centre will basically be working on this same project, this molecular fingerprinting.
This will be one of the core activities. That's correct.
Okay, great. Now, on a slightly different theme, I also wanted to ask you about an initiative that you and your colleagues founded in 2022 called Science for People. Now, this is a charity that aims to provide education for children in Ukraine who have been displaced and disrupted by the war. Can you tell me about this organization and how you became involved in this?
Thank you for the question. Yes, the direct motivation for this initiative came from the very sad events in Ukraine shortly after the war in Ukraine broke out. I thought one way or the other we should try to help, and after some thinking, I concluded that perhaps the best way I could potentially help would be to try to reach out to the community that I know well, to my friends in the scientific community, and try to ask them for donations with which we would try to provide in the best possible way help to young people suffering directly or indirectly from the war within Ukraine. Many of them left the country, as we know. Millions of them left the country, and many of them are living here in Germany, and they are being taken care of. But there are also very many millions who had to flee from their homes but didn't leave the country. Many of them actually settled in Western Ukraine near the Hungarian border. And that's where we teamed up with an organization, a local organization taking care of those refugees, and developed programs for supporting them. One of our flagship programs is what we dubbed Scola Plus, which is a tutoring, extracurricular tutoring program, where in the framework of which we pay teachers, local teachers who give extracurricular tutoring to both refugee children as well as local children. And now ever more schools are joining in. Meanwhile, we have reached out with this program even to a couple of schools near the front line, and the program is expanding. And we try to extend this kind of support to as many schools as possible, of course, depending on the donations that we can collect.
Okay, so you're basically providing sort of extra tutoring for children that have had their education disrupted.
Exactly.
Yeah. And is this just within science or all areas of the curriculum?
It is basically essentially all areas, right? It's up to the schools and the teachers who are supposed to actually assess in which subjects the pupils do need this kind of extracurricular support most, and that's basically their decision.
Okay, and so you said you're using it'll be like local teachers in Ukraine that are doing
Exactly. So
yeah.
we basically do the two good things in this way. On the one hand, we support the teacher who are not very well paid in Ukraine, particularly under the current conditions, and provide them a kind of additional incentive not to flee the country but stay there and help the needy. And secondly, these teachers, with our help, provide help to the children whose parents don't have to pay for this. So this is free for the children, of course.
Excellent. Well done. That sounds like a really fantastic initiative. That's great.
Thank you.
And then finally, as part of Physics World's coverage of the International Year of Quantum Science and Technology, this year we're asking all our interviewees to briefly tell us, what does quantum physics mean to you?
Well, I would say quantum physics is the area of science which describes phenomena in the microscopic world. We physicists had to learn about 100 years ago that the laws of nature appear to be very different in tiny microscopic dimensions at the atomic scale as compared to the laws that govern processes that we can observe just directly at a macroscopic scale. And that's, I think, what quantum physics is about, to actually establish these laws, and with the help of those laws describe microscopic phenomena. So this is, I think, the main mission of quantum physics. But quantum physics actually does give us much more. Actually, it opens completely new areas also for very exciting revolutionary applications that may change our life, like quantum computing is a completely new kind of computing which opens entirely new opportunities. But actually, even what we have talked about just in the last few minutes is kind of a consequence of quantum science, that we can actually probe with light the molecular composition of human samples, and can learn details about the health state of an individual, is at least indirectly a consequence of quantum science, and is a kind of exploitation of the phenomena that can only be described by quantum physics.
Excellent. Well, thank you very much. Thanks for your time and for speaking with us today.
I thank you very much for the possibility. Thank you.